So I'm talking about Flaviviruses. So what's classified as the family Flaviviridae, actually, which is the button, the red. Yeah, okay. Actually consists of several genera. All right. So probably the most famous over here are what's called the genus Flavivirus, which are mostly insect or arthropod-borne viruses, a group of mosquito-borne viruses, tick-borne viruses. There is a small subset which has no known vector, but still fall within this classification. And this is most of what we're going to be talking about today. But within the family, there are a couple other, or two other major genera, which are the Hepatitis C viruses, which I'll try to touch on a little bit near the end of the presentation. These are blood-borne, but not carried by mosquito vectors. And also the pestiviruses, which tend to look a little more like HCV. These also not generally insect-borne. And we're not really going to talk about these today, but many of them are fairly important pathogens from a veterinary perspective. So a lot of these are actually major diseases of livestock. So within the family, there are certain commonalities that apply to all these viruses. They are positive sense RNA genomes of about 10 to 11 kilobases. They're all enveloped. They, all of them encode a single open reading frame. So it's, from start to finish, it's essentially one long protein product, but that protein product is cleaved into functional subunits by a combination of proteases that are present in the host cell, as well as protease that's encoded by the virus. And we'll go into that in a little more detail later. So as I said, we're probably going to focus mostly on the mosquito-borne Flaviviruses. Yeah, I don't know. The mosquito-borne Flaviviruses shown on the bottom, including and that group includes things you may have heard of, like yellow fever virus, West Nile, dengue virus, Japanese encephalitis, and sort of most recently or most popularly Zika virus. I think I can get. Okay. So the general outline of the lecture is just going to go through some history and discovery of several members of the Flavivirus family, as well as associated pathogenesis. Then sort of moving to a more general overview of the Flaviviruses, we'll touch on structure and all the highlights of the life cycle, including entry, replication, translation of the protein, assembly, and release. And then with all viruses after that, the whole thing starts again. So the word Flavivirus comes from the Latin word flavi or flavus for yellow, because the first virus sort of described in this this group was yellow fever virus, which was known long before the days of molecular biology, going back to the 17th and 18th centuries. And it used to be quite a problem, not only throughout the world, but in the United States, including up along the eastern seaboard, much further north than we ever think about yellow fever occurring now. And in this particular page that I copied out of a 1793 account of a description of yellow fever, you can see, and the most notable thing is their weird predilection for using the letter F in place of S. I don't know what's up with that. But you can see that the description of the disease is pretty horrifying, characterized by most notably the black vomiting, which is actually hemorrhaging and coagulated blood into the gut, which then is vomited up. Liver damage, notable by the yellowing or jaundice of the affected individual. And it was also observed that in many cases, the disease had this biphasic characteristic where there would be initial fever, which generally gave way to what appeared to be recovery, followed by a relapse, which was characterized again by hemorrhage, shock, and in many cases, death. Right? So as I said, it was a major problem early in the century because it tended to be brought up every season by ships, a lot of it stemming from the slave trade. And mosquitoes would be transmitted even to areas with a cold winter where they would not stay, so it was somewhat seasonal in the disease would come out during the summer when the mosquitoes were able to leave the ships, and then they would set up shop near the docks. But back in those days, pretty much everybody lived in that area as well. So this was sort of not only a major problem within the colonies in early Americas, but throughout the Western Hemisphere, and it's also present in Africa as well. So it was initially attributed, or early reports of it tend to attribute things like bad air, miasma, which you see in lots of 18th century discussions of disease. But in the late 1800s, a Cuban scientist named Carlos Finley hypothesized that it was actually mosquitoes that transmit this virus. And this hypothesis was looked at extensively over the subsequent years, and most notably tested and proved by Walter Reed and some of his colleagues, including Jesse Lazier, in the early parts of the 20th century. And this was done by taking people who had yellow fever and letting mosquitoes feed on them, and then letting mosquitoes feed upon naive volunteers. Actually, probably one of the more salient data points was obtained by Jesse Lazier himself, who fed one of the infected mosquitoes on his own arm, and he contracted yellow fever, and then he died, which is generally how we don't do this kind of thing today. But they were able to show that indeed the mosquito is required for transmission of the virus, and not only that, there's one of the observations that Lazier had made was that there's a period of incubation that has to occur in the mosquito. We now know this is replication of the virus in the mosquito gut, so it's not merely just transferring the virus from one individual to another. There's a replicative cycle where the mosquito takes a blood meal from an infected individual who has virus circulating in their blood. The virus replicates in the mosquito and then is able to pass it on to a naive individual. They also did all sorts of other cool, further controlled experiments to rule out other mechanisms of transfer of the virus, like having more volunteers, sleep in the unwashed beds of people who had just died of yellow fever. So cool stuff like that. But this led to, knowing that preventing yellow fever, at least before there was a vaccine, which I'm going to talk about in a few minutes, but you can combat it by controlling the vector. So mosquito netting, getting rid of breeding grounds for mosquitoes. This was also, I guess, most famously instrumental in allowing the building of the Panama Canal and things like that. So this is what I've alluded to earlier, the infectious cycle of yellow fever virus. So after infection of an individual, there's a period while the virus replicates, and then what we call viremia, which is circulating infectious virus in the blood. At this point, the person is able to transfer virus to a mosquito if the mosquito takes a blood meal from that individual. Then there's the extrinsic incubation period, in which the virus is replicating in the mosquito gut, at which point then the virus is present in the salivary glands of the mosquito, and it can be transferred to a naive individual when the mosquito feeds again, and so on and so on. So a lot of the viruses I'm going to talk about today, yellow fever, dengue virus, Zika virus, these are in fact primarily transmitted from human to mosquito. And some of them also have probably non-human primate reservoirs that also allow the virus to maintain itself in certain environments. Other members of the Flavivirus family, like West Nile virus, which I'm not going to talk about very much today, are generally considered dead-end hosts in humans. So a human can be infected and there is occasionally symptoms, but viremia in that individual is never very high, so it doesn't result in transmission of the virus to other mosquitoes. In the case of West Nile, that generally happens in birds. So humans in those cases are not part of the normal infectious cycle, but in some cases do get bitten and can contract the disease. Good, all right. So then in the early 30s, Taylor and Smith came up with this idea of, or developed the yellow fever vaccine. So it had been known, since Jenner, it had been known that for certain viruses, once you're infected with it, you are then immune to subsequent infections with the same virus. This was known to be true for yellow fever. And they took an isolate of yellow fever and passed it through chicken embryo cells sort of continuously for about 3 years. That's 200 or more passages through the cell line. And they found by testing in monkeys that the resultant virus was no longer particularly pathogenic, but it did provide protection from subsequent infection with pathogenic strains. So this was effective as a vaccine, and it's still more or less the vaccine we use today. And we know that there are differences from the pathogenic strain, about 67 nucleotides and 32 amino acid differences. But the actual mechanism of attenuation, why it's less pathogenic, is, I think, not fully defined. There are a couple theories, some that maybe there's just less diversity within the vaccine strain, i.e. it's not like a swarm of many different viruses. But this, I think, still remains to be proven.
A couple questions?
Yeah. Oh, sorry.
Are the same nucleotide differences seen with every attenuation sequence? I mean, are you seeing the same genomic changes that cause?
Not necessarily. So you could probably do this several times, but what we use as, the yellow fever strain 17D, which is the vaccine strain. So that's now been sequenced, and that's where you have the 32 amino acid differences. You could probably repeat this experiment, get an attenuated virus, and it's likely it wouldn't have all the same changes. Although maybe some of them would be conserved. That wouldn't surprise me too much. Yeah.
I know you said it's not fully, the mechanism isn't fully understood. Is there any postulation about if the attenuated virus, if it's allowed to reproduce, or if it's given, an embryo to reproduce in, if there's less of an immune response, or if it doesn't have to, if it's allowed to mutate so that it doesn't have to maintain those pathogenic genes?
Right. So generally, the idea of attenuating a virus through passage in cell culture is I think exactly what you're suggesting, that if you take away its need to combat host immune response to grow within the host and just sort of make it focus on growing in tissue culture, it'll adapt itself to the tissue culture, and that's not actually optimal for the host. So that's how you kind of drive it to the attenuated state. Is that what your question was? Yeah. despite the vaccine, the virus is still a threat through much of the tropical and subtropical world, shown here in yellow within South America and Africa. And I believe recently, maybe in Angola or elsewhere in Africa, there have been severe outbreaks of yellow fever that generally send the local officials, public health officials, scrambling to try to vaccinate the local populace, which can be quite an effort for some of these countries where infrastructure and available resources are quite low. So I seem to remember having read then about this recent outbreak that then they had to prevail upon other countries who had reserve stocks of yellow fever vaccine to ship them over to the affected country. So this remains quite an issue in many parts of the world still. Yeah.
What is the vaccination rate in those areas?
I actually don't know exactly what probably different countries in different parts of the world may attempt to vaccinate most of their population, but I don't know. I'm sure in many of them it's probably quite close to zero, but don't quote me on that. But
I've been following this a little bit, and it does seem to be pretty minimal. It's been rather lax, and it does seem to be, as Alec mentioned, it's really mostly about the mosquito vector control, which seems to work much better than the vaccine.
Right. I should also say that the virus is not absolutely attenuated, and it is not recommended for certain people, especially those who are immune compromised, which in certain areas of sub-Saharan Africa, given still the prevalence of HIV, can become something of a problem.
With the yellow fever vaccine, do you have to get vaccinated every year or something like that, or is it just one time?
It's generally one time, and I think the actual, those who have followed the immunity over time shows it's pretty long lasting in most cases. So this is the principal vector for a lot of the viruses that normally grow in humans. So at least for yellow fever, for dengue virus, for Zika virus, Aedes aegypti is the major culprit. And it is very much evolved to live in close proximity to people. It's an urban mosquito. It's out in the day. When people are out, it preferentially likes people. And it can breed in very minute amounts of water that are left around in any sort of, small depression that'll collect water in any sort of city or urban environment. So it's difficult to get rid of. The secondary vector is Aedes albopictus, or the tiger mosquito. This also is out in the day. I didn't make this slide, but the author means little c Catholic in terms of not, applying to lots of different, all-encompassing. So humans and other species, it applies to all of them. And you find it in both the rural and urban environments. These mosquitoes, the albopictus, are capable of transmitting these viruses, although often, or at least in the case of dengue, the epidemics tend to follow the range of Aedes aegypti more than they do of albopictus. Just a quick note on mosquito control. So, as I said, this was realized in the early 20th century that mosquitoes were the vectors for a lot of these diseases, and mosquito control, either through getting rid of sources of standing water where mosquitoes could breed, or probably large-scale insecticide spraying, was undertaken, sort of vastly reducing the ranges of the mosquito, although that has sort of lapsed, and aegypti has come back to most of its previous areas, at least in the Americas. Yeah.
Is it because whatever was being done, like the mosquitoes evolved?
some resistance, some of it being just the 70s kind of correlated with a lot of authoritarian fascist type regimes in South and Central America who could spray insecticides without really worrying about what the populations thought about it. And we have a little less of that now. Not to advocate for these regimes or anything like that, but so a combination of just sort of changes in how mosquito control is managed with some resistance amongst the mosquitoes, and then once you give them some space, they kind of very rapidly will repopulate their previous areas, since their host, their food is people, so it'll just show up where the people are. Yeah.
I've always considered, or I had a factoid in my brain, I don't know if it's correct, but yellow fever really does stay in the tropics, between tropic cancer and tropical cancer born.
Now that's pretty much true. I mean, so obviously it didn't used to be when the mosquitoes were brought up. So a lot of
Well, I guess I'm just curious, like, is there something about temperature that will keep, I mean, obviously the mosquitoes going outside the tropics, but is the temperature of that, both those ranges do something?
right. So the winters outside those ranges will kill the mosquitoes. So in the case of the Philadelphia epidemics of the 18th century, that was continual reintroduction every summer. But they don't live through the winter outside of these areas.
The virus itself wouldn't be affected by a temperature change?
Well, the virus has to live either in the host or the mosquito. So it's sort of staying with those. So once the mosquito vector dies, people who had yellow fever either recover or they die, in those days, and then it went away for the winter time.
Yellow fever doesn't affect the mosquito though, right?
It doesn't kill the mosquito. It does, in fact, infect and grow in the mosquito. So
Does it kill the mosquito?
no, it doesn't kill the mosquito. So, yeah.
Is there like another organism involved, like within the mosquito?
In certain non-human primates, if that's what you mean. Oh, you mean within the mosquito?
Well, like in the gut of the mosquito, is there like a symbiont?
No, it grows in mosquito cells. There are some current getting a little outside my field here, but current strategies of virus control of giving mosquitoes these symbiotic bacteria which help prevent virus infection. But that's not, normally the virus is replicating in the cells of the mosquito and in the cells of the human host, or in the forest cycle in the monkey host. Good? Yeah, all right. Okay, so we're going to hit one of the other major flaviviruses, dengue viruses, which are actually really a set of 4 different viruses or serotypes, which are defined by their antigenic differences or binding and ability of antibodies to neutralize them. So there's 4 types, dengue 1, 2, 3, and 4. It's generally known that if you're infected with one of these, you have lifetime immunity to that same serotype, but not to the other serotypes, although there is a short-term cross immunity following infection. All the serotypes cause severe or fatal disease, so they're all classified as dengue because the disease looks similar, and they are closely related viruses. Within each serotype, there's genetic variation, so it is probably true that within each serotype there are more pathogenic and less pathogenic strains, although this is hard to prove because in any particular outbreak you have questions of the genetics of the populace as well as the particular variant of virus that's showing up. Previously, the virus disease had been classified as dengue fever, which is a severe febrile syndrome, and then it could progress to the more severe forms like dengue hemorrhagic fever, which included vascular leakage, and that could potentially go to dengue shock syndrome, which is actually a potentially lethal form of shock due to fluid loss through the vasculature. This is, yeah?
Are the serotypes area specific?
They tend to be. I mean, but now they're constantly sort of shifting. So generally at any one time, one area might have one serotype predominate.
Okay,
If
so
that's-
it's not like the multiple serotypes in like one infection?
Well, now we actually see that too. It used to be more like, there would be one serotype present, and maybe after some time it would be somewhat replaced by another one as there would be some introduction into that area. But there are currently now areas in Brazil and Southeast Asia where there are probably multiple serotypes circulating at once. Yeah.
Since there are only 4 serovars, is there a vaccine that covers all 4, or is each vaccine very specific to?
Well, I'll talk about that in some detail in a few slides, if that's okay. All right. So currently, just to say for anyone thinking of doing tropical medicine in the future, the current classifications are dengue without warning signs and dengue with warning signs, that is, warning signs of potential hemorrhagic disease coming up later, and severe dengue, which is the hemorrhagic manifestations. This was done largely because the individuals with severe dengue can be treated if they are admitted to a hospital, and they'll generally survive if they are given constant care and fluid management, basically managing the shock. But again, admitting people to the hospitals in a lot of the countries with endemic dengue is a big strain on resources, so a major question when presented with a dengue case is who can be sent home because they will most likely recover, and who needs to be watched for onset of severe dengue. So management of cases is a big question, or a big emphasis of those who actually treat the patients.
Are you, if you're sick, hemorrhagic and mosquito bites you, are you still virulent to others? No, we had somebody with dengue and a few mosquitoes in the room, shouldn't we all run away?
Actually, the viremia precedes that a little bit. So probably by the time they're in severe distress, they're unlikely to be infectious to the mosquito. I don't have that. There's a slide, which I don't have in this particular deck, that shows, the viremia tends to coincide with the fever, but not so much the hemorrhagic symptoms, which come later. So this is the range of dengue as we've seen for yellow fever. It's similar, although dengue extends also into Southeast Asia as well. And again, just between, largely, around between the tropics of Cancer and Capricorn is where the major range of the mosquito and the major range of the virus as well. But it does occasionally make forays up into the United States. This is from the CDC's weekly report. This is in 2009 and 10. Some mosquitoes set up shop in an area of Florida, and there were dengue cases. The U.S. normally sees several of these diseases, mostly travel-associated, people who were visited endemic countries, but once in a while you get infections from people who didn't leave the United States, because there has been some transmission from local mosquitoes through the local populace. And we've seen this since this time happen periodically, usually in Florida, sometimes southern Texas with dengue virus, and also most recently with Zika virus.
Do the hemorrhagic symptoms, unless you're getting to that, do they follow the symptoms of fever pretty closely?
A few days. Not in all cases, though, only in certain ones, which, well, I'll deal with that right now. So what we know is that, so as I said, one serotype is generally thought to confer lifelong lasting immunity to that same serotype. And much of this data, even defining what isolates belong to what serotype, was done again by human trials. A lot of these, the seminal studies that we still have most of our knowledge of about, times post-infection to various aspects of disease, come from work by Albert Sabin, the oral polio vaccine guy, which he did during World War II, and published a lot of these studies in the early 50s. And he observed that initially following dengue infection, generally the individual is immune to probably all the different serotypes, and then that wanes after about 9 months, where you're left with just immunity to the one serotype that infected you in the first place. The second observation is that secondary infection with a heterologous serotype. So if you're infected with dengue 2 and then infected with 1, 3, or 4, the potential for severe disease is increased. So generally you see more severe dengue and hemorrhagic manifestations after a secondary infection with a different serotype. And that led to this hypothesis championed by a guy named Scott Halstead and others of antibody-dependent enhancement. So the idea here, and this can be a little confusing, so stop me if it doesn't quite make sense, but the idea being that you have circulating antibodies against, say, dengue 2, and those will react to the other dengue viruses. Well, we know all this antibody interaction things were all worked out. This was the first way of identifying viruses before there was any sequencing. So just interaction with serum from somebody infected with one virus, if it really interacts, they have the same virus or it's slightly related. But anyway, so there is interaction with the viruses of different serotypes, but they're not very effective at neutralizing. So if you coat dengue 1 with antibodies elicited from a dengue 2 individual, that virus is not killed. It can still infect cells or mice or whatever your assay method calls for. So the antibody-dependent hypothesis is that we know there are cells in the body which bind to antibodies and take them up, as well as whatever's attached to it. And maybe that if you have a sort of bound but not neutralizing antibody to one, you get greater infection of some of these cells, like macrophages and endothelial cells, which results in more severe disease because more cells can be infected. Does that work for people, for the most part? Okay.
What did you say came up with the theory of antibody?
It's generally attributed to Scott Halstead, Halstead, and if you ever meet him, he will tell you about it at length and tell you why he deserves a Nobel Prize. Yeah.
So if you have antibodies, but not for that specific serotype, a virus, then it can actually increase
Right.
the severity of the infection because it maybe masks the virus or allows it entry into these other cells.
That's
that would typically kill it.
That's exactly right. Right, and so then we were asked before about vaccines, and do you need a vaccine against one or all of them? Well, this has extensive implications for vaccine development because you pretty much have to vaccinate against all 4, because if you just create antibodies against one, you will protect against that one, but you're potentially making infection by other viruses worse. Yeah.
Are there cases of people being infected by more than one serotype at the same time? Or is it because they're protected for that 9 month period?
not too aware of any. I mean, there's probably some examples, but not much that I could talk about that would be at the same time. But you generally see it. I mean, it can be over a period of years that you're subjected to another serotype and get more severe disease. So through that 9-month period, you're probably, after you've had one, you're probably good against all the serotypes for a few months before those other protective antibodies tend to wane.
Is it possible for one serotype to mutate into a different one while inside the host?
No, I mean, they do, there is drift amongst the serotypes in terms of sequence differentiation, but you generally don't get so much that you're antigenically different like that, or at least, well, not over, sort of relatively short periods of time. I mean, the 4 serotypes probably did evolve from a common ancestor at one point, and so over tens, 100,000 years, or I mean, for as long as the virus has been around with humans, I guess it has been co-evolving, and probably the different serotypes come from geographic isolation of the different populations of viruses. So they did probably evolve into different serotypes over evolutionary time, but not within a single cycle of infection. So some experimental data which shows that the antibody-dependent enhancement phenomenon can be modeled in mice. So wild-type mice are generally not susceptible to dengue virus because of aspects of mouse innate immunity, which the virus cannot overcome, but these are done in interferon-deficient mice. Just a caveat about mouse models of dengue infection. But if you infect mice with dengue 2, this is actually a dengue 2 virus which has been adapted to mice. They infect these interferon-deficient mice, they die. That's shown, these are all the same data points in these open triangles. So this is survival. You infect the mice with dengue 2, and they die, pretty much the last one going in about 11 days. If you pre-treat that mouse with serum from an animal that has received dengue 2, so you get some protection of those animals because the virus binds to and neutralizes, the antibody binds to it and neutralizes the virus, right? But if you give them serum from animals that have been infected with dengue 1 or 3 or 4, the death actually occurs much faster. So the disease is worse. So this is pretty much in agreement with the hypothesis of what could be happening in humans as well. And you can see here by just these curves here indicate that the serum binds to these viruses in all cases. But these are the neutralization curves. So here you generally do this by diluting your antibody and seeing how far can you dilute the antibody and still prevent infection, in this being in tissue culture cells where you can look at viral plaques. And you can see in the dengue 2 case you can dilute pretty far and still get reduction of up to half the number of plaques in a tissue culture dish. Oops. But with the other ones, they're not significantly different from just an unimmune sera sample. So the neutralization of the virus is very important for the protection, but binding antibodies that don't neutralize exacerbate the disease. So let's see, running a little bit behind, but just a quick synopsis of Zika because everybody loves Zika. So Yes. So this was, so just a quick thing. Zika virus was first identified in 1947 by a group of scientists working for the Rockefeller Foundation and Imperial College of Medicine in Uganda. And they were studying yellow fever, actually. And what they had done was taken rhesus macaques. Those are not native to Africa. They're imported from India in this case. And they would put them in these towers and keep them in a cage on top of the tree and look, just check them every day and see if they got sick, hoping to sort of look at infection with yellow fever. But they also picked up a lot of other viruses. So one animal was found to have spiked a fever one day, and it was brought to the lab, and they took serum from it. And were able to pass that serum on to baby mice, which actually are very permissive for lots of viruses. They don't have much in the way of immune systems, and this was 1947, and that was the tissue culture dish of that time. So they had this virus, which they could pass through suckling mice. And they also were able to isolate the same thing from local mosquitoes. And once they sort of determined the characteristics of an antibody response, they saw that, in fact, the virus seemed to be fairly prevalent throughout Africa, in that many people had antibodies that recognized Zika virus. Later on, it was identified also in Southeast Asia as well. So people were aware of it. It was this antigenically distinct viral entity named Zika after the forest in which it was discovered, which is just a map showing you an idea of where it is, which is right here. And a lot of the RNA and mosquito viruses that we study today were all, or even ones that we've heard of but don't pay any attention to, were discovered by these guys during this period of time. But up through the beginning of the 21st century, there are maybe a handful of confirmed Zika virus infections, and these were mild fever-like cases in which the individuals always recovered. So it was not a big deal and not something people spent a lot of time researching. I just added this slide in just as to give you an idea of, like, when you talk about discovering this virus or this virus, how do you know it's yellow fever, or how do you know it's Zika virus or dengue virus back in the 1940s and 50s? And this is again done all by serum binding. So how far can you dilute your antibody and still get reaction or inhibition of hemagglutination in this case? And if you can dilute it really far, you suggest that this is the same virus. And then you can try to group viruses into related families based on it. You can dilute it part way and still neutralize the virus, but not as far as what seems to be the same virus. I mean, and it's kind of a, nothing is that there's going to be differences based on the individuals where you got serum from, if you're working with human serum, or the strains of mice or strains of viruses that you have. But they did a pretty good job of actually mapping out how these viruses relate to each other. So these down here are all Flaviviruses. These are all alpha viruses. As we know now, so they're now classified as different families. But before, they were just things you could get from a mosquito, but through antibody analysis, they were able to tell kind of what's related to what. And this has all been borne out by sequence analysis today. So sorry for that brief interlude, but so the spread of Zika virus. We know that it had been in Africa, it had been in Southeast Asia. It was not a big deal until about 2007, when it showed up in the island of Yap in the South Pacific, and this was associated with a large outbreak of symptomatic disease, fever, muscle ache, things like that. None of it was fatal, but looking into this epidemic, it was determined after ruling out dengue and chikungunya virus and yellow fever, that going back to old studies and viruses, they found that it was in fact Zika virus. And then a second introduction into French Polynesia in 2013 was not only accompanied by a large outbreak of fever and muscle ache, but also Guillain-Barré syndrome, which is a partial paralytic disorder which occurred after infection, and in retrospect also a certain number of birth defects associated with infection during pregnancy. They hadn't noticed that initially in French Polynesia because the numbers were so small, but they went back and looked after the virus got to Brazil, and we have the explosive epidemic that was described in the news for quite a while. And this, so when it first got to Brazil and spread through South and Central America, we saw not only the increased number of symptomatic infections, but also more of Guillain-Barré syndrome and a large number of birth defects, which seem to, current numbers suggest that maybe 5 to 10% of pregnant women who are infected with Zika could have some sort of, about 5 to 10% of those cases could result in neurological defects in the developing fetus.
Guillain-Barré syndrome is occurring in the adults that were infected.
Sorry, yes, that's an adult, somewhat autoimmune disease. It's directed by antibodies that recognize certain proteins on peripheral nervous cells. And it has been seen with other viruses that do this, or that where recovery is then associated with Guillain-Barré syndrome.
Epstein-Barr is Epstein-Barr?
No. So, open question still about Zika virus. What changed after 60 years? We knew about it. It was circulating. Nobody cared because it didn't really do anything. But then suddenly it shows up. So is the strains of virus, did it evolve somehow to become more pathogenic? We don't really have evidence of that yet, but it may be the case if we look in the right way. Is there a difference in its interaction with the host, or is it merely that you introduce this thing into, say, Brazil, where there was a large population of Aedes aegypti and a naive population, so it was able to spread very quickly, and the numbers made it appear very severe, and are we going to sort of then move into a situation where it's more of a constant endemic but smoldering infection without the sort of startling numbers of symptoms coming up? We still don't know. Another possibility is that it has something to do with co-circulation of other viruses like dengue and chikungunya virus, which we know are present in Brazil, and throughout most of the region where we've had the Western Hemisphere epidemic. And there's a lot of interest has been given to, can this antibody-dependent enhancement phenomenon apply to dengue, from dengue antibodies to Zika? So there's some evidence yes and some no so far. So I think we don't really have a good answer to that yet. Is there a question?
I'm just going to ask about the co-circulation. We're talking purely about co-circulation in terms of the vector. We're not talking about like a satellite virus kind of situation where it needs dengue or it needs the virus to replicate.
It doesn't need it to replicate, certainly. Does it need it for causing birth defects? Well, we don't know, but I mean, it's possible that could have some effect, again, probably through this antibody interaction that we know occurs in the dengue viruses. And then how does it cause microcephaly or other birth defects? That's also an open question. People have been trying to address it in a bunch of different ways, using in vitro growth assays in neuronal progenitor or placental cells, mouse models of infection, as well as non-human primate models of infection. I don't know how well you can see this here, but this is just some data taken from a mouse paper. The point is just here, so this is, these are placental cells, and for the sake of just making the point, the green is, healthy cells. And in the case of Zika virus in these mice, so again, wild-type mice will prevent Zika virus infection, but if you disable the innate immune response through knocking out the interferon receptor, you get more disease and quite a destruction of a lot of different placental cells. And then if you look over here, I don't know how visible this is, but maybe you can see some red dots in this region, or not, but trust me, they're there. And that's just a measure of apoptosis, so programmed cell death of fetal neurons in a Zika-infected fetal mouse brain, whereas in the controls, there's a certain amount of this that goes on, but much less. So whether or not the mouse model is a viable model for human disease is also, I guess, a point of some contention, but it probably does provide some valuable information for figuring out what's going on. All right, so now, Jesus, sorry, I probably got a little bit ahead of myself because you've asked a lot of good questions, but all right, let me just go quickly through the Flavivirus replication cycle. So as I said, so we have entry. It's an envelope virus, so it gets into a compartment in the cell and then still has to access the cytoplasm. So that is done via a low pH dependent fusion with a late endosome, which delivers the nucleic acid into the cytoplasm. Then, as I had mentioned, you translate a single polyprotein, which is cleaved into multiple subunits, into the functional proteins of the virus. It uses so it's a positive sense virus, so that virus served as the mRNA for translation. And then it also serves as a template for a virally encoded RNA polymerase, which makes a negative strand copy down here. And then that negative strand copy is a template for making more positive sense RNAs, which are then packaged into the virion, and the virion then moves through the secretory pathway. There's a maturation step at the end, so it needs to encounter the host protease furin, and then it gets out. So the structure of these things, this is the envelope. It's not the capsid, right? So the envelope proteins lie in this regular icosahedral shape with each protein sort of, they form dimers and lie more or less flat against the surface of the membrane. The inside, the capsid and nucleic acid is, as far as we know, fairly unstructured. So it's not that protein capsid that you would see in something like an adenovirus, with a clear icosahedral shape. This is the outside of it forms this shape. Just the schematic of the envelope dimer. So you can see these are two separate envelopes, one here and one here, lying anti-parallel to each other. So entry is affected, as I said. It requires pH reduction as you move through the endocytic pathway. So you bind to a receptor. The receptor not actually known for any of these things, or in some cases people have suggested them, but they don't sort of hold up in every situation, so we're not sure if it's somewhat promiscuous in terms of receptor usage or how that works. But it does go in through receptor-mediated endocytosis, which results in lowering the pH of the subsequent vesicles. When it gets to the late endosome, about pH 5.5, there's a conformational change in the envelope protein, which allows fusion, just focusing on the entry pathway, which allows fusion with the membrane here and release of the nucleocapsid. So a sort of schematic of the fusion, you have the dimer lying next to each other at low pH. The dimer, they rearrange into trimers, which form this sort of spike conformation here with the fusion loop shown here in orange. So in this example here, just take a look. So you initially have the dimers lying against each other. The low pH conformational change, which form these trimers, which then have transmembrane, the initial transmembrane domains in the viral membrane, as well as the fusion peptide, which inserts into the host membrane. And then, again, through conformational changes, that draws the two membrane segments together, resulting in the formation of this fusion area here. I'm just pointing at my screen. That doesn't help you. So that the viral nucleic acid can move through this section. Okay, I've sort of screwed up on time, so I'm trying to move quickly through here. So this is the polyprotein. So as I said, one reading frame. The front end is the structural regions, the capsid, the envelope, and PRM is sort of another membrane protein which acts to some extent as a chaperone for E and prevents premature fusion. If you look at the membrane topology of the thing, it's translated sort of in and out of the ER membrane. So the capsid is made in the cytoplasm, and then the back end of it is translated into the ER membrane where you get the envelope proteins. So remember, the ER lumen is more or less contiguous with the outside of the cell, the way the secretion machinery works. And then you have this set of other proteins, NS1, also primarily ER luminal, as that it's a secreted protein. And then the rest of them are either membrane associated or largely outside of the membrane. So shown here in the little black stars, these are cleavages made by ER resident host protease. So as this is made, the host itself makes these cuts, so you get these separate proteins separating from the polyprotein. Right? On the other side, NS2B and 3 together will make their kind of auto cleave themselves from each other here, and then they'll make other cleavages here to get the rest of the functional subunits. So we have both the virally encoded protease as well as the requirements for cellular ones. Not all of these proteins are fully defined as to what they do. NS5, we know, is the RNA-dependent RNA polymerase, and it also encodes a methyltransferase, which is required for putting a cap onto the genomic RNA so you get proper translation. I'm going to skip this one. So back to the flavivirus life cycle. When you're actually forming the replication cycles in, when you're actually forming the replication complexes in the cell, it results in this crazy rearrangement of the cellular membranes, which, excuse me, I just turn this off. Where the replication happens, and a lot of EM work has gone into sort of defining how this all works and developing of these 3D structures and models where the RNA is replicated on membranous surfaces in these replication complexes, and it associates with the capsid protein, which then buds into the ER, acquiring the envelope as it goes along. So if you'll sort of look at there, so there's the replication complexes and the translation, and then sort of budding into the ER lumen, you have the virus, which then mostly what you have are these sort of vesicles which carry multiple viruses through the secretory pathway to the outside of the cell. And when I did this last year, I kind of ran out of time with hepatitis C, and I guess I'm doing it again, but I think I should have 5 minutes till 10:05. Okay, so Sorry, I hope you liked the first part before I ran out of time. So just hepatitis C virus is medically important, and it's a member of the Flavivirus. It's a little different than the mosquito-borne things. It does not have an insect vector. It is purely blood-borne. And it was known for some time when they knew about hepatitis A and hepatitis B as a leading cause of hepatitis that was non-A and non-B. It is a disease characterized by generally a mild acute infection, but a predilection for for establishing a chronic infection, which was a lifelong thing that had to be dealt with that could often lead to cirrhosis and liver failure. Liver cancer, for a while, was one of the major causes of required liver transplants throughout the United States. It was known that this was a viral agent, filterable agent. They were able to pass it to chimpanzees. Generally, we don't do any chimp medical research anymore, which is probably justified. But if there was here's one case where it may have been justified. I'll leave that up to you. But there's no other susceptible animal model, and it also has the distinction of being the first virus sort of discovered by purely molecular means, in that it was just cDNA from RNA taken from infected individuals was used to express little pieces of protein, and then that was those cDNAs were probed with antibodies against the virus, giving you sort of a piece of the virus sequence that allowed the researchers to sort of walk across the genome and determine the whole sequence of this thing. But once they knew the sequence, that allowed screening of blood products, which had been the major cause of transmission of hepatitis C, and then taking them out of the nation's blood supply, preventing further transmission, although still we see new infections largely based on IV drug use. And the rate of humans who are still dealing with chronic infection worldwide is still about 3%. So that's actually pretty big when you think about the population of the world. So as I said, same general architecture as the mosquito-borne flaviviruses, a little bit different. There's no cap. It has an iris at its 5' end, and it has 2 enveloped glycoproteins instead of 1. But the general replication strategy still applies. Single polyprotein with the structural regions at the front end, and a variety of host and viral proteases which are required for cleaving the individual protein subunits, and then replication through a negative strand intermediate. Another interesting sort of intersection between scientific and social research right now is the fact that there are now very good anti-HCV drugs that have been developed largely by Gilead, a pharmaceutical company down in San Francisco somewhere. Previously, HCV had been treated with just giving exogenous interferon or ribavirin, drugs with limited efficacy and fairly nasty side effects. But a new class of drugs specifically targeting the HCV polymerase, NS5B, and also this is now packaged as a combination therapy with an NS5A inhibitor, have been made available, and these are very effective in just wiping out the virus from the infected individual. But they are incredibly expensive, so in order to avoid liver cancer, you come across with something like close to $200,000. So $200,000, or you die, or whatever.
What's the significance of 12 and 24 weeks?
That's generally how long it, so not entirely. So this is your slide, Ken, but I believe the first 12-week course takes that long, and sometimes it takes another 12 weeks to fully get to absence of all detectable virus. So they'll treat for as long as it takes, but generally that's the full course of
So does
infection.
HCV infection count as a pre-existing condition?
Right, so insurance coverage of these drugs as well as, people's ability to pay is a major hurdle. On the other hand, it did cost the company a lot to develop the drugs, so I don't know.
Isn't there a vaccine?
There is no vaccine against HCV.
So let's thank Alec.